Attention deficit hyperactivity disorder, or ADHD, is a neuropsychiatric disorder that affects impulse control and other cognitive functions within a person's brain. In the general population, 3% of adults are affected and 5% of children, with ADHD being more common in boys than girls. ADHD primarily begins in childhood and is characterized by several key traits. Inattention, hyperactivity, and impulsivity are all characteristics of the disease. Further, more than 2/3 of individuals with ADHD experience coexisting conditions, among which include insomnia, mood or anxiety disorders, and substance abuse. Dozens of gene changes are associated with an increased risk of developing ADHD, with many of these thoughts affecting the development of the brain. In addition to genetic influences, approximately 10 to 30% of ADHD risk is thought to be influenced by environmental factors such as chemical exposure and nutritional factors, among others. Although there are a wide variety of genes as well as external factors that impact the risk of ADHD, the two specific genes that we'll analyze for the test are the BDNF and COMT gene. We believe these two genes best highlight ADHD risk. The brain-derived neurotrophic gene BDNF provides instructions for the production of the BDNF protein found in the brain and spinal cord. The BDNF protein is a neurotrophin, which are proteins in the brain that impact the survival, development, and function of neurons. The protein of neurons by facilitating the growth, maintenance, and maturation of the cells. The protein also helps regulate sympathetic plasticity, which is important to learning and memory. The catechol O-methyltransferase COMT gene is responsible for the production of the COMT enzyme. This enzyme plays a role of deactivating catecholamines, which are mixtures of adrenaline, hormones, and dopamine that are released when the body is placed under stress. The enzyme also breaks down neurotransmitters in the frontal lobe responsible for short-term memory, executive functioning, and behavioral inhibitors. We plan on including 8 genetic variations that affect ADHD susceptibility on our SNP chip test. The two SNPs that we think best show the risk of ADHD include the RS6265 SNP as well as the RS4680 SNP. The RS6265 SNP is found in the BDNF gene. The genotypes for this SNP include homozygous A and G alleles, AA and GG, or the heterozygous AG genotype. The A allele encodes the methionine amino acid MET, while the more common G allele encodes the valine acid VAL. In studies published on the effects of the altered BDNF gene, it's been shown that youths and adults with the VAL-MET or heterozygous AG genotype were more susceptible to ADHD-related symptoms. The homozygous AA genotype was linked to a variety of other diseases such as Alzheimer's, while the GG genotype did not affect the gene. The BDNF gene, the G allele, is substituted by the A allele in codon 66, the BDNF protein, creating the AG genotype that reduces the secretion of BDNF and effectively increases inattentive and hyperactivity symptoms, which are both components of ADHD. The RS4680 SNP is found in the COMT gene. The genotypes for this SNP, similar to the RS6265 SNP, include AA, AG, and GG. The G allele likewise encodes for the valine amino acid, while the A encodes for the methionine acid. In the AA genotype specifically increases the chances of ADHD. According to a study examining the effects of methionine allele on RS4680, children possessing the heterozygous AA genotype or 2 of the MET alleles perform significantly below average on tests of sustained attention. Because the production of the COMT enzyme is slowed down by the altered RS4680 SNP, dopamine is broken down slower in the frontal lobe, affecting cognition. This further affects executive function, leading to ADHD-related symptoms. Our test could help individuals be aware of their current or future risk of having ADHD. Being knowledgeable of this information can immediately help individuals look for and be aware of resources that can help them deal with living with the disorder. Being aware of ADHD can also help in preparations for future costs for treatments such as medication that can help ease the disorder. ADHD, as stated earlier, when testing as a precaution and give insights into symptoms and behaviors that are exhibited by varying individuals. It does not paint the whole picture. Other genetic variations that are not accounted for in our test could also play roles, as well as environmental factors. While our test could show that an individual has a high risk of ADHD, they could be ADHD-free going forward and could go on to live healthy lives due to their setting in a healthy, stress-free environment. In contrast to this, other SNPs that aren't as well studied could also play definitive roles in the chances of getting ADHD. ADHD can be expensive and worrisome, which are some financial and emotional factors to consider. Although the disorder is not life-threatening, dealing with someone who has it or being someone who has the disorder can be an emotional experience categorized by confusion or fear. Test results may also result in individuals facing stigma or even experiencing a placebo effect. As for financial concerns, since this disease is not life-threatening, some people might find the benefits of undergoing the genetic testing minimal when compared to the cost of the test itself. Further, while the test itself is minimally invasive, it is significantly more invasive than the DSM-5 analysis, and so subjects may choose to simply wait until symptoms present themselves to get a completely non-invasive diagnosis. Thank you.